Okay, now in this video we are going to be examining the tricarboxylic acid cycle, abbreviated the TCA cycle. This is also known as the Krebs cycle, named after Hans Krebs, who discovered the biochemical reactions that occur in this cycle, or it's also called CAC for citric acid cycle. I will refer to this as the TCA cycle. Now, in order to start the TCA cycle, we have to have our substrates. And the first substrate we're going to be looking at is a substrate we generated using pyruvate dehydrogenase. And this is, of course, acetyl-CoA. The acetyl-CoA was generated from pyruvate, which came down the pipe from glycolysis. Now recall, we are in the matrix of the mitochondria. Mitochondrial matrix. And all of these reactions in the TCA cycle will occur in the mitochondrial matrix. So, first principle to realize is that this is a cycle. So we are going to have a start point, we are going to go through several reactions, and then we are going to return to the same place that we began. And where we're going to begin is examining the entrance point of the TCA cycle. This is a 4-carbon intermediate, and it is called oxaloacetate. So the first reaction of the TCA cycle is a condensation reaction. In other words, we are going to be creating a new carbon-carbon bond, and this is a condensation reaction to attach the acetyl-CoA onto the oxaloacetate. Precisely what we are going to do is be adding this carbon onto the number 2 carbon of oxaloacetate. This reaction is done by an enzyme called citrate synthase. Now the standard free energy change of this reaction equals -7.7 kilojoules per mole. Notice the large negative free energy change, so this indicates that this is a very favorable reaction, and it's going to move forward and not be able to reverse. So the reverse of this reaction is not biochemically possible in the cell. Where the energy comes from is the hydrolysis of this carbon-sulfur bond. Always remember that when we have acetyl-CoA on the carbonyl, this is a high energy bond. So the breaking of this bond is going to be the energy that drives this reaction. So the product that we make of this reaction is going to be a 6-carbon TCA intermediate. Here's our 6 carbons. 1, 2, 3, 4, 5, 6. So this group here, this acetyl group, came from acetyl-CoA, and it was attached to the carbonyl of oxaloacetate. Because the enzyme is named citrate synthase, we are going to synthesize citrate. So this product is called citrate. This, incidentally, is where the name citric acid cycle came from, the first product entering the TCA cycle. Now, the next reaction is a very reversible reaction, and it will depend on substrate-product relationship, as we've seen our free energy calculations before. And what we are going to make here is a modification of our 6-carbon intermediate. We're moving the hydroxyl group down one carbon. This particular intermediate is called isocitrate. The enzyme that performs this reaction is called aconitase. Being readily reversible, we are going to have a delta G 0' that is pretty close to 0. In this case, it's positive 1.5. So what we are simply doing is moving this hydroxyl group down to this carbon. We are moving the hydrogen up here. So you can imagine that if we're moving an OH and an H, this is going to be involving a dehydration and rehydration reaction in order to move these functional groups. Now the next reaction from isocitrate is going to be an oxidative decarboxylation reaction. So what that particularly means is that we are going to be losing a CO2 molecule, and we are going to perform a redox reaction where we are reducing NAD to NADH. So the carbon dioxide that we're losing is this one right here. It's in the middle of the chain. The bond that we are going to be reducing, reducing those electrons to NAD to make NADH, is right here, this carbon-hydrogen bond. So after performing this reaction, we will generate a 5-carbon intermediate. So again, we've lost the CO2 decarboxylation reaction, and we have reduced this hydrogen-carbon bond, generating a carbonyl. Now the enzyme that performs this reaction is called isocitrate dehydrogenase. To perform an oxidative decarboxylation reaction. Now, as in other dehydrogenase redox reactions, this is going to be an irreversible reaction. So the reverse of this reaction is not going to happen. And that is primarily because the standard free energy of this reaction is a negative 5.3 kilojoules per mole. So this large negative delta G is being driven by the redox reaction, and that makes it a one-way reaction. The product that we are forming here is called alpha ketoglutarate. Important to know, we've lost a carbon. We started out with our 6-carbon intermediate, and now we're down to a 5-carbon intermediate. Remember, ultimately this is a cycle, so we have to get back to 4 carbons. So the next reaction is going to process alpha ketoglutarate, and in processing this reaction, this is going to be a decarboxylation reaction. So we are going to lose CO2, and like the previous reaction, we are going to have a redox reaction accompanying this. What will we form, because we're losing a carbon, is a 4-carbon intermediate. Now on this carbon, which is a carbonyl, we will have bound to it a succinyl, an acetyl-CoA. So that means that the decarboxylation reaction we're performing, we're not going to lose these two electrons right here. And so these two electrons will be transferred onto NAD to form NADH. We also have, to balance our equation, the introduction of a coenzyme A in order to add on to here. Now recall, this being a thiol group, carbonyl bound to sulfur, this is a high energy group. It's a high energy bond. This is called succinyl coenzyme A, or succinyl-CoA for short. The enzyme that performs this reaction is alpha-ketoglutarate dehydrogenase. And as you can imagine, like the previous dehydrogenase enzyme, this is going to be an irreversible reaction. The delta G prime 0 equals negative 8.0. So the reverse of this reaction, again, is not possible. Now the next step in our reaction, we're getting closer to returning to oxaloacetate. The next reaction, we are going to generate, of course, we're maintaining our 4-carbon structure. We are going to be converting the succinyl-CoA into succinate. So the important reaction here is that we are losing the bound coenzyme A. We are hydrolyzing this high energy bond. We are going to utilize this high energy bond to transfer the energy to another energy storage molecule. In this case, we are not talking about ATP, but we are talking about GDP. And of course, a coenzyme A is released from this reaction. So we are simply transferring the energy from this high energy bond of the thiol group onto generating a high energy phosphate bond on GTP. Now this enzyme is called succinate thiokinase. Now you might think, because there's energy involved in this reaction, that the delta G, 0, the standard free energy, is going to be very high value, but actually it isn't. The delta G 0 prime is actually negative 0.7. Close to 0. So that indicates that the reverse reaction is very, very possible. This is simply due to the fact that we are not gaining or losing energy, we are just transferring energy from this high energy thiol linkage, transferring it to the phosphate linkage to make GTP. Recall, GTP can be used in protein synthesis. So now let's move to the next page and continue processing our TCA ingredients, in this case, succinate. So here is our succinate molecule. And we are going to continue processing this until we get to oxaloacetate. So the next reaction here is going to be another redox reaction. And we are going to be generating a double bonded carbon in the center. This is succinate. This is called fumarate. Now notice what we've done here is we've generated a double bond between the two middle carbons. This is a redox reaction because in order to do this we have to remove 2 electrons along with a hydrogen. Now, if you recall when we talked about FAD and NADH, this particular reaction, to create a double bond, we are going to be removing a hydrogen and 1 electron from each of these carbons. These are single electron transfers, and so they are going to be transferred not to NAD, which requires 2 electron hydride transfer, but rather to FAD to make FADH2. This enzyme, of course, is a dehydrogenase, and it is called succinate dehydrogenase. The standard free energy of this reaction is equal to approximately 0. So that means this reaction is readily reversible. Now, the sole reason we are preparing this is for the next reaction. We are creating a double bond here in order to prepare it for the next step, which is hydration. So in this next step, we are simply going to be adding water across this double bond. Here's our water across this double bond. This molecule is called malate. The enzyme that does this reaction is called fumarase. Because this is a simple hydration reaction, the delta G 0 prime, standard free energy, is also, like the previous reaction, approximately 0, making this reaction readily reversible. Now, if you recall the structure of oxaloacetate and compare that with malate, you'll see that we are now only one step away from generating our initial substrate to begin the TCA cycle. This will be done by the enzyme malate dehydrogenase to get us to our final step. And our final step, of course, is oxaloacetate. So in order to transform malate into oxaloacetate, we are going to simply oxidize this carbon. And we are going to be grabbing these 2 electrons and turning this alcohol into a ketone. So in order to do our redox reaction, we need a place to put the electrons. And of course, we are going to place those on NAD to make NADH. Now, this enzyme has a very, very unique standard free energy. In the forward reaction, the standard free energy is a positive 7.1. Now, based on what we have been discussing before, this reaction in the forward direction, you would think that this reaction could never, ever possibly go, because the reverse reaction would be a negative 7.1. However, in this particular case with malate dehydrogenase, it comes down to our substrate product relationship. So remember, our true free energy calculation is not just the standard free energy. it is going to take into consideration the natural log of the product over the substrate concentration. So recall, if we have very little product and a lot of substrate, we have a very small number. The natural log of that number is a large negative number. Multiplied by the gas and temperature constants, we will have a very large negative factor to give us a negative delta G. Now the question is how do we keep the product of this reaction low enough that we can overcome this unfavorable standard free energy? The way we do that is we introduce acetyl-CoA. Now recall, acetyl-CoA is continuously being generated by pyruvate dehydrogenase. From pyruvate. Pyruvate is coming from glycolysis. So as long as we are oxidizing glucose in glycolysis and pushing acetyl-CoA through degenerated by pyruvate dehydrogenase, we will have the substrate to form the condensation reaction with oxaloacetate to form citrate. So it's the abundance of acetyl-CoA that is coming through from glycolysis that keeps oxaloacetate concentrations very, very low. Keeping that low will drive a negative delta G to keep this reaction going. Now one more note to make in here is the enzyme succinate dehydrogenase. This is a very peculiar enzyme because it is integrated into the inner mitochondrial membrane. All the other enzymes in the TCA cycle that we've looked at are soluble in the matrix. This enzyme is embedded in the inner mitochondrial membrane, and it's done so for a very important reason. The transfer of electrons to FAD occur in 1 electron transfers to FAD. So we are making from FAD to FADH, then we are adding 1 more electron to make FADH2. Whenever single electrons are added, single electrons are very dangerous. They have to be controlled, the movement. And so the FAD is permanently bound to succinate dehydrogenase. So that means in order to return to the oxidized state, in order to keep the enzyme going, we have to transfer these electrons off into the electron transport chain. The electron transport chain is embedded in the inner mitochondrial membrane. So the succinate dehydrogenase is embedded in the membrane precisely to locate it adjacent to the electron transport chain, so that when it oxidizes its FADH2, these electrons go directly into the chain, the process, regenerating FAD so we can keep our TCA cycle going. So this is the only unique enzyme in this manner of the TCA cycle. Succinate dehydrogenase is embedded in the inner mitochondrial membrane, whereas all the other enzymes are located in the mitochondrial matrix.